Hello again. This is the first of 3 videos in this particular series dealing with the topic of DNA functions. This video is about replication. The next 2 will be dealing with protein synthesis and mutating. The fact of the matter is that DNA has to be able to replicate. We always represent DNA as looking like this. And we have to clear up some terminology a little bit here. This is a chromosome. Each one of these strands is a chromatid. So this one, and also this is a chromatid here. So a single chromosome consists of 2 chromatid strands. If we consider all of the chromosomes in a cell, in the nucleus of the cell, there's our cell, here's the nucleus. Inside here, however many chromosomes there are, if it's a human body cell, we've got 46 chromosomes, but that is the chromatin. Okay? So the terminology, similar terms, but they mean different things. Chromatin is the genetic material in a cell, all of the DNA. A chromosome is a strand which is made up of 2 chromatids. Now, when people see DNA, they don't see it looking like that because DNA is typically unraveled and it's just too small to see. But with a light microscope, anyway, with the light microscope during mitosis, you can see DNA and it looks like this. So it'll be lined up with the equatorial plate. Let me get rid of this. And if this was a cell that had a couple of chromosomes in it, they'd be attached to spindle fibers like that, and those would be 2 chromosomes. 2 chromosomes. A chromosome has 2 chromatids. This is a chromosome, but there's actually 4 chromatids making up this chromosome. That's what we have to talk about. DNA has to be able to replicate. It has to make copies of itself because during mitosis, one of the copies has to go to each side so that you wind up with the correct genetic makeup in the daughter cell. So if we have a 46 chromosome cell dividing, you have to have 46 chromosomes in each of the daughter cells. So we have to make copies of it. How does it work? Well, we have to go back to this picture here, not this one, and consider the process in a little bit of detail. Now we know that this chromosome has got hydrogen bonds in between the complementary bases. If this is going to replicate, it has to be able to copy all of this stuff. And what happens, quite simply, is there's an enzyme called DNA helicase. DNA helicase breaks the hydrogen bonds in between the complementary bases. So what happens is, I'll just draw it a little bit smaller here. If this is the DNA strand, these hydrogen bonds are still intact. These are not intact, they're just fragments of them. It breaks the hydrogen bonds between the complementary bases. That's the function of DNA helicase. So it breaks the H bonds between complementary bases. Now, I'm going to come up with a little bit of a base sequence here. A, T, C, G, G. I've got an extra space in here, hang on. G, T, C, A. We know what the base sequence on the opposites, on the complementary strand is because these things are always in the correct pairs. So that T would be bonded to that adenine, This T would be bonded to an adenine there. This would be guanine. This would be cytosine, cytosine. adenine, guanine, thymine. So that's what happens. That's the first thing. This happens during interphase in preparation for cell division. The second thing that happens is complementary nucleotides have to be moved into position here. I'll erase this and I'll follow this with the second thing. Remember the story about the nucleus, and the nucleus has the nuclear pores in it and things can get into the nucleus? Well, we have nucleotides which are assembled out in the cytoplasm out here, and inside here is where the DNA is actually replicating. So the nucleotides have to be moved into position. That's the role of a second enzyme. The DNA helicase breaks the bonds. This one, DNA polymerase, the DNA polymerase is a group name for enzymes. There's lots of different DNA polymerases. This one in particular moves new nucleotides into position. Now, when I say into position, I mean into their complementary position, because we know where they're going to go. We know how they're going to bond together here. Now, if I take new nucleotides, I can suggest to you right away that this one has to be a thymine nucleotide. This one has to be a guanine nucleotide. This one has to be an adenine. This one is cytosine. This one is cytosine. This one has to be an adenine, cytosine, thymine, guanine. These are the nucleotides, the new nucleotides that get bonded into position. And they form hydrogen bonds. So remember, there's 2 hydrogen bonds here, there's 3 hydrogen bonds there, there's 3 hydrogen bonds there, there's 2 there, and so on, because of the dipoles which line up properly. That's step 2. The third thing that happens involves a different DNA polymerase. And what happens quite simply is once these are in the correct position, DNA polymerase bonds them together to form another strand. So bonds the new nucleotides into a strand. So we have this happening. This one then bonds to this, to that one, to that one, to that one. They all bond together like that. And this process continues all along the strand. So it starts at one end and it proceeds. So DNA helicase comes along and it breaks it as it goes up like that. And as that's happening, this part's being put together down here. We call this first thing here unzipping. Because it's kind of like a zipper that unzips, so it just goes sequentially along the chromosome. One thing that is very important to notice is that these strands are identical now, aren't they? Just look at the bottom. AT, CG, TA, GC, GC, and it would be all the way up like that. So it produces 2 identical chromosomes. I better say they're identical chromatid strands, because that's what they are. They're still connected in here. So because they're connected, it's still a single chromosome. They're not considered separate chromosomes until they're broken apart. So they are identical chromatid strands. And also, worth noting is that each of these chromatid strands that gets produced has got an old strand and a new strand. So the old strand, the original strand, is the one that's in black, and I used a purpley color to represent the new one. So these strands are what we're going to say is half new. The process is called semi-conservative. To represent the fact that it's partially new and partially old, so it's conserved the original DNA sequence that way, or the original base sequence that way. Now all of this is occurring before mitosis starts. So what we had at the beginning of this video is a little picture of a chromosome in a cell looking like this. What this is, quite simply, is, I'll try and represent that. It's a duplicated chromosome where each strand is, that's these two strands, each one is half new, half old, it's still one chromosome. They're held together by a little piece of DNA called a centromere. And you can see the semi-conservative nature because I've colored it purple and black. And these are attached to the spindle fibers at the centromere, and the spindle fibers contract, the centromere breaks, and the new cell will have an identical chromosome here, an identical chromosome on that side. Now, during mitosis, what happens to the nucleus itself, and what happens to all the organelles? Because if you look at these cells, this is all you really see in cells which are dividing. The fact is that the nuclear membrane sort of retracts. They say it dissolves. It doesn't really dissolve. It sort of retracts. It's continuous with the endoplasmic reticulum, that endomembranous system, and all of that membrane material actually goes to the outside, to the periphery of the cell. So it's just inside the cell membrane, and that leaves lots of room in the middle of the cell for the chromosomes to do their separating like that. Once it's all happened, once these are all in the separate cells, they can begin to wrap up around the histones again, and you won't see them. They become that, the double helix strand that we would represent them with otherwise. So, like I say, this is the first of 3 videos dealing with DNA functions. The first one is the necessity of DNA to be able to make replicas of itself, replication, and the purpose of that is so that daughter cells can have identical chromosomes following cell division. Thank you very much.